In this video, we will discuss about tuberculosis pathogenesis. So, what actually happened in the tuberculosis? Tuberculosis is an airborne disease. It spread uh, from one individual to another individual through air. And in tuberculosis, what happens is that it affects the lungs. Usually affects the lungs, but it can also affect the other part of the body, such as brain, spinal, kidney, etc. etc. So if tuberculosis remain untreated, it can even cause or even lead to death of the patient. So how this tuberculosis cycles works is we will understand this from the figure. First, the bacteria, that is M. Tuberculosis enters into the host through air, and it will bind into the lungs, or we can say uh, bacilli inhaled as a droplet and then binds into the alveoli of the lungs, and the first primary infection occurs, and that will leads to activation of immune system within the patient, and it will clear out this infected macrophages. And however, what happens is that it will not be able to clear out all the macrophages, and there is some uh, latent latent infection occurs. That means that uh, se- uh, uh, this virus, that is M. tuberculosis, continues to grow and develop within the host patient. However, it is in dormant state, and after some time it reactivates itself. And after reactivation itself, before reactivation, initial form is known as granuloma formation. As we can see that the M. tuberculosis or infected macrophages are surrounded by the cells, so that it will not be able to uh, recognize, or it will not be able to recognized by the immune system, or will be able to hide themselves from the immune system of the patient. And after some times the granula become mature, and this mature granula will leads to uh, further infection to the patient, as it we can see it. And this infection will cause again the activation of the adaptive immune system. So by that time, immune system of the body gets uh, aware about the antigen of this affected tuberculosis, or we can say M. tuberculosis antigen of, is aware about the, or trained about to act against the M. tuberculosis antigen. So adaptive immune response will get activated when the latent infection gets mature and become active. And in some cases, what happens is that this granular breakage occur, or we can say caseating granula occurs, and this leads to spread of this virus across different parts of the body. As it we can see in this, it is spread across different part of the lungs. And when this granula gets caseated, it will spread across different part of the body with the help of the air. And this, when infected person come in contact with, or is there is a possibility that this infected person can infect other healthy person with the help of or through air, as it is, as this tuberculosis can be spread across individual through airs. And the figure on the right hand side describes various strategies that can be used the to identify the condition of this tuberculosis cells with the advancement of the flow cytometry. And here some of the examples are shown, such as in order to check the metabolic activity, we can use fluorogenic probes, so that it will bind to the specific enzyme, and we will be able to know whether that specific enzyme is present or not, such as esterase dehydrogenase. and other probes such as live cell probes, or that can be used to check whether the cells are alive or not, or that probe are also there to check whether the tuberculosis affected cells are dead or not. And antigen responsive, in order to check whether the antigen is uh, present in the tuberculosis, we can use antigen responsive fluorescent antibodies. So if certain kind of motifs are expected to be present within the or on the cell surface of this tuberculosis, we can check them through the with the help of antigen responsive fluorescent antibodies. That is, it will only give fluorescent signals when it will bind to the epitopes for which it is designed for. And another example that is shown here is that fluorescently labeled peptides, so that it can bind to only those kind of proteins or enzymes that have the sequence for which that has the sequence of peptide for which this fluorescent fluorescently labeled peptide is designed for. So from this we can find out whether a particular protein of interest or enzyme is present within the cells or not with the help of flow cytometer, by designing the peptide in a way that can only that can bind, that is specific to the protein of interest. So all these things are the examples, and we can see that earlier it is difficult to do analyze the tuberculosis, uh, the cell, the analyze the tuberculosis cells, as the tuberculosis cell envelope is made up of multi layers, and it is difficult to penetrate the tuberculosis cell envelope or cell membrane. However, nowadays strategies have been developed, so, by which we can the penetrate the protein of interest or fluorescently labeled peptide within the cells. Some of the examples are like lentivirus infection or transient transaction, stable transactions, by which through such strategies we can inject the designed fluorescent and can observe the behavior in the flow cytometer. I hope you have understood something about how this tuberculosis has spread and how people or researchers are used the flow cytometer to identify more in-depth knowledge about these tuberculosis infected cells. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.